Come closer.
Okay.
So we have all the researcher for us, and this is a unique opportunity. So don't be shy. I mean, it's the time for sharing and for asking.
And I will start with one very easy question. And I'm looking to every one of you. When will we know if this is a dominant negative or not? When?
Allora,
Not if, when? So who start?
Yeah, so, you know, again, Stefano generated the schematic. I'm not entirely sure where he got 12 to 18 months from. But yeah, as it relates to Argentinian 497, I hope as soon as possible. I would like to think that some of the assays we described are pretty feasible within that window, maybe sooner. Yeah, I thought one of the more impactful messages I heard, which is not at all to be pessimistic, but was Katarina's point that dominant negative and haploinsufficiency, I think, could depend on CHAMP1 function. It kind of seems, especially as we get more researchers involved, CHAMP1 is kind of this jack of all trades, protein and gene, and really has its kind of fingers in a lot of different pies. So that is to say, you know, I think the good news is a lot of these gene therapies, a couple of which have been described, we can use them in combination with each other, at least in theory, right? So even if it is dominant negative, even if it's not, right? Even if, you know, ultimately, I do think that the issue is we just need the right dosage of functional CHAMP1 protein for any and all of its functions. So as soon as we can sort of figure out that Goldilocks, yeah, I think we can implement it. But hopefully, yeah, some at least positive or negative data as it relates to Arginine 497 from our work pretty soon. So.
Thank you, Zach. New name, Katarina. So it's,
Yeah,
yes,
I
it's
mean,
not an interrogation, but of course, every one of you will speak. So take care.
you know, with the disease, you want an answer. You want an answer now, and you want one answer, right? Because you want one cure. Unfortunately, in biology, it isn't so easy. And again, if I make the analogy to a lot of cancer genes, the reality is that any one protein has more than one function. And the reason is because those proteins need to communicate with each other, right? Through organelles, between cells. And so different mutations may affect different functions more than others. And we need more research. We just need more research.
Fine.
okay. An easy question for all of us, for all of you, or who wants to answer. What do you bring back to your laboratory from these 3 days that we had, if any? Who want to respond?
Otherwise, we pick.
Well, I would just say to try more. To
Try
just
more.
try more.
Thank you. Let's do like that. 2 words, everyone.
Try more.
New collaborations.
A general comment. First, I was convinced that haploinsufficiency cannot explain CHAMP1 disease. So I hope you are convinced now that loss of the entire protein is not the same phenotype, because loss of the entire gene is associated with a very mild phenotype. But thinking about a complex protein-to-protein interaction, maybe that loss a specific domain or a specific site for interaction can be the key. So not just haploinsufficiency, half protein normal in its structure, but loss and imbalance of specific domains. I'm rather inclined to think about that. So experiments I realized that are going on from all point of view, clinical, therapeutic, and also on basic researches. So I do hope that we can reach new important insight in next close future. And
Very
this
good.
is my final message. I'm convinced about that.
I guess I will take back that we really need all the models, I guess.
I'll say, for me, I was interested in the point mutations, which are largely not the focus of research. I think there's more research on the level of protein and on the truncation. So maybe I will try and look at the point mutations, because that's something that biophysically we can do a rigorous job on.
I'm going back to your question about what we're taking home from this conference, and I link to what Stefano said, more collaboration, and I stress again what I also said during my presentation this morning, and I think I can speak in behalf of Professor Gasparini as well. We're coming from...
Paolo.
Well, this is our first approach to CHAMP1 because we're coming from the White Satin Syndrome study, and we've been following the last year White Satin Syndrome. So last year conference was my very first approach to CHAMP1, and what I took home last year, and I confirm I'm taking home this year too, is this need for a collaboration and to look at the two diseases as a whole, so implement the research not only in White Satin Syndrome, so in our original field of research, but also in CHAMP1.
Tanta roba.
Hi. I take home these fantastic families and the desire to understand better the phenotype and the correlation. It's very useful, this moment, all together with other researchers.
Yes, me too. It's wonderful to see all these children, because give us the opportunity to see the entire phenotype that we have to still understood. So everyone can give her part, his part in this project. Thank you to you.
So for me, what I bring home is, as a person that just entered into the CHAMP1 field, for how much I can read and study, interacting with all of you and seeing all of your researches and experiences is an invaluable thing. And, you know, interacting also with the family gives me a new drive to, you know, apply what I've learned in these days and bring it back home with me and try to do better. That's all.
Yeah, I couldn't agree more with everybody up here. I'll stick to the two words. I'll say bigger picture.
I would say that we all have to do more, and I will bring back that, like, of course it was wonderful to be here and listen to what people do and see families, but we will apply to more grants, including every possible foundation, because NIH kind of gave up on us. So we've been submitting grants, but we're gonna do more and like literally try every opportunity, because if you don't ask, you don't get.
I bring home a lot of energy here, a lot of person that wanna do a lot of things. Family are very united, and my point of view is not a point of view of a scientific researcher, but a gestional. And I think that to gain a great results, you need a lot of research, and to do a lot of research, you need to be a good structure, and this is what I take to you.
And for me, let's say I'm bringing home two things. First of all, as medical geneticist, I had the opportunity to have a look, despite I was not visiting your kids. I paid a lot of attention to them. I look at them, and now I am much more convinced as before coming here that once we will got the approval, part of them could be enrolled in the clinical trial, and most likely with positive effect. It's something we shared also with Anna. This is the first point. The second thing is that you should, in some way, because families are always looking at therapy, and the good thing I've learned is that the three major areas of therapeutical interventions are here represented, because there are scientists working on gene therapy, and there are scientists working on NO1, ASO, RNA therapies, and we were talking about drug repurposing. So despite this is a small community, the future of the therapeutical intervention is already here. We don't know which one will be successful, but this is something also good for all of you, because one eye on the three major areas is present.
Thank you very much.
So the
very
most
inspiring.
common wording I heard was collaboration.
Yeah.
That's like, we really think that it's what we can bring to use us, for sure, before closing, but we're not really closing, but before closing, for sure, use us in whatever way you need, as we are going to ask us, and yeah, and families too, of course, ask. So once you are asking, we can move questions, and we can progress and go on together. Is there any other question? Yes, please. I want like to go to bed at 3 a.m. like 2 days ago.
Thank you. Thanks to all of you who've contributed. This is my first time I've come to this meeting, and yeah, I've been really impressed by the work that's going on. I had no idea that we had a couple of small molecules in development, potential for gene therapy. So I think that's really exciting. I'm going to ask you a question that I know none of you are going to be able to answer, but I think that will be on the minds of most of the parents here. And the first one is, when will we actually get to a therapy that can be used clinically? And a question that's specific to my position. My son is 20 years old. Will there be a sort of a cutoff age at which therapy perhaps will become less effective, bearing in mind the sort of the development of the brain and nervous system, sort of tails off maybe mid-20s, I think. Thank you.
$1 million question.
So I'll answer the second question first, which the hope is obviously no. But I think that, unfortunately, to answer it kind of indirectly, I think that is the importance of still understanding the mechanism, right? So, and mechanisms. It's not simply, I mean, of course, as soon as we have a putative therapy that works, I think all of us would, there's no competition here. The word collaboration is obviously out and about. So really the first available therapy that seems effective is what we would go with, right? But in terms of the cutoff age, right, I think that's when we need to understand the mechanisms at play, both throughout the course of neurodevelopment, and then obviously are those mechanisms that we could also alter later in later stages of life, right? So that is to say, selfishly, as a basic scientist, as a researcher, that's the value of doing the basic science and really investigating some of these molecular and cellular mechanisms. So I know it's not a very clear answer. I wish I had a more clear answer, but that's what I would have for that one. So if anybody else has insights.
Honestly, I think the first drugs are likely to be approved drugs repurposed, because they give you the most immediate way to get things into clinical trials, which will allow you to have access to the drug. But those may not be the final drugs that give you the best benefit or the best benefit to your child, or the ability to reverse some of the damage. Those may come, unfortunately, later.
Yeah, I mean, I agree. In terms of timing, it's clear everything is related to the difficulties and the availability of the drug. So clearly, drug repurposing, it's the most close one, and not only in perampanel, maybe other drugs. It's clear that in this case, it should be something like a symptomatic drug helping in solving some issues like behavioral issues, but you are not curing 100%. This is genetic disease. To cure, you should completely replace the gene not working with the right one. In this light, you asked how many years. The timing to get even what they are called the advanced therapeutical medicinal product, the ATMP. So gene therapy, cell therapies are categorized as ATMPs. The time depends on the funds, but it depends also on the technology. The technology is improving a lot. We didn't mention today genome editing. Still, there are issues with genome editing, which means instead of going around with just to exactly change the wrong mutation even in vivo. There are some side effects right now, some issues to be still discussed, but the technology was not available a few years ago. So probably in the next 5-10 years, there will be much more opportunities for all the patients affected by rare diseases. According to, I fully agree with Zach regarding the second part, when it will be the best window and until which age. Of course, in principle, sooner is better. That's clear. But for example, in our case, we got some positive results that we are thinking to make another amendment and start treating also patients with White-Sutton syndrome with older, I mean, let's say up to 40 years old. And nobody knows if it works or not. Probably for some specific signs, it will work.
And yes, that's what we like.
Yeah, I just want to make the analogy again with another, it's not considered a disease, but it's aging, right? Where we do get cognitive decline. And we know that intermittent fasting or caloric restriction actually can improve the cognitive awareness, even like until very, very late in age. So I think it's also important not to give up hope and not to give up, believe in science, if that makes sense. It may not be completely reversible, but it may definitely be improvable.
Yeah, it's probably just some fine tuning on that question, because I was anticipating maybe for a drug to help with development, and somehow, as you said, the earlier the intervention, the better, and it would somehow amplify development. But that you mentioned a perspective on maybe even reversing effects is very new to me. So I wouldn't have expected that possibility. You think that might really be possible to change some?
Again, in aging, the whole caloric restriction field is actually pretty impressive, and it's impressive because it's an on or off. So in life, when you turn caloric restriction on, you start to live longer, and then you turn it off, and you start to live less longer, and then to turn it on again, and so it's really an on and off switch. From a scientific point of view, we know a lot already how this works, but by far not enough. But it's just one example where it's possible.
Yeah, specifically with trichothiodystrophy, which is, say, mutations in ERCC1, there were children who couldn't walk or talk or stand, and by doing caloric restriction, they were able to walk and ride a bicycle and speak. So that's very much a reversal. Of course, they're young age, but I think the hope is if you can rebalance things so that the systems can work in concert rather than fighting, there is a hope to see improvement,
I had some more comments, very, very simple. Of course, it's hard to think about the possibility to restore completely the neuro developmental phenotype. However, to my opinion, we have a very good points at this moment. The first one is the absence of major brain malformations. And the second one, we noticed that CHAMP1 disease is not a regressive condition such Rett syndrome is, for example. And each child I met again this conference improved on support therapies. So they are good starting point. And finally, we are aware about the plasticity of neurons. So, of course, we could think about improvement on many aspects, mainly behavior abnormalities, but maybe some more else.
Thank you very much.
Other question? Well, I have one question for you. How do you think we, as an association that is spread off worldwide, but, Alliance, yeah, well, which is spread off worldwide, but is also very tiny. How can we make not just the public aware of CHAMP1, but the researchers, because we need more people researching about CHAMP1. So do you have any advice for us because we have to move in several directions. As you see, we are quite a little but focused group of people. And how can we be more focused on this?
Before anyone replying, no, suggesting to give out free beer is not an option because you already did. So that's it.
So let me say, first of all, you mentioned before it was a cost here. Of course, it was a cost, but it's an investment.
Of course.
More than a cost.
Otherwise, we wouldn't do it.
Yeah, because the possibility of having all together, it's probably will give you in terms of revenues.
Yeah.
Say, much more than the cost per se. The second important point is, in my opinion, is that research is important. It's clearly important, but sooner or later it should be focused in some way, because otherwise, and us as a physician, we should also try to do something, even if we don't know 100% what is underlying a disease, because otherwise we will stay stuck in the middle of nowhere. And in some way, we should also take our risk and our responsibility. And so, and focus should be, because otherwise the research is very, very expensive. And there is another, in my opinion, important point that you should take in consideration, which is not only the public awareness, but is to force in some way the stakeholder, in Italy are the politicians, to to in some way consider that this disease, as well many other rare diseases, need to be considered completely different from the other diseases. This means investments from the public institutions, but also means to get benefits for the patients. For example, in Europe, there is a kind of healthcare system that can support a lot of families and patients. And this is not easy. Definitely is not easy. And there is a possibility also to join other charities like Teleton or Geneton in France to go direct and combine for a research call. And this could also help because they have the possibility to reach a very broad community of scientists. And that's it.
Yeah, I cannot agree more with Paolo in terms of focusing the research. However, thinking outside the box, for instance, interfacing with a larger community of rare diseases might attract the attention of other people that are studying probably completely different diseases, but with unsuspectable overlapping mechanisms. So my point is, if you show up at a congress in which many other rare diseases are talked about, someone might have the solution in their hands, even if they don't know, and maybe because they are studying it for another type of problem, but this might apply to CHAMP1. So my solution is interfacing with other broader communities while staying focused.
I agree completely with you. We need to talk about CHAMP1, and we need to go to congresses. Two, actually, yeah. We need to talk about CHAMP1 congresses and so on. We need to reach out as scientists, right? We need to send students, postdocs, and so on to conferences to talk about our project, and I think that one is important. It's a kind of awareness, right? For for CHAMP1 syndrome, it is very important.
I mean, just to tag on to this, I was made aware of the foundation and of the research by Alan DeAndra wearing a CHAMP1 t-shirt. That's how I got here.
That you have now too.
That I have now too.
Cool. So.
Try our beer. Where's beer? The last Congress, we had free beer for everyone.
To me, I think one way to draw scientists and attention is to increase the amount of data, because machine learning and large language models benefit. The more, say, full genome sequences for the more patients and families you have, the more that people can say, that's interesting, how can I relate this to all the other sequencing data? There's just a lot of RNA-seq and DNA-seq out there. The larger your Alliance database is, the more families, the more variation and commonalities, similarities and differences people can find and relate to behavior. That becomes incredibly compelling, because genes that influence behavior and development, that's a big future area for neuro research. And if you say, we have a window, a special window, we have all this data, that will get people very interested, I believe.
We are collecting some good homework here.
I just wanted to add off the back of sorry. No, I was going to say off the back of what you said, Stefano. If you are going to conferences, you're sending postdocs or students to conferences, and you feel having a patient advocate, having one of us there would be beneficial. Ask more of us. We're more than happy to travel, be involved in any way we can. We're a resource that we're more than happy to provide whatever we can to support you. So
I think patient advocates are huge.
Yeah. Massive. Yeah. I think just to sum up a lot of what everybody's saying, for you guys, and I think that the CHAMP1 Alliance, all the foundations already do a really great job of this, but just continuing to championing the science itself, right? So advertise, whatever breakthroughs we sort of have, any sort of work that we're publishing, right? Because, I think clinicians and scientists alike, they like solving mysteries and solving puzzles, right? So, and I think, that idea of having a phenotype and having some sort of phenomenon that you really can assess, right? There's so many unknown players in neurodevelopment, also just disease. Right now we have people that investigate the metabolic aspects as well as the DNA breaks and everything. So yeah, the more you just push the science out there, I think the more people you will attract, and that's true for the data as well as what's coming out from you guys.
I have a very pragmatic thought. Build a scientific committee that is very that is work and enlarge all of the network and stabilize to grow in all of the world, and knowing the scientific research.
Yeah, I wanted to ask, because you mentioned it briefly, if there are any hopes that AI technology will somehow accelerate everything, the process.
I see Thomas that want to answer.
I think AI is going to be one of these things that'll either be the greatest thing ever, or it'll be a big bust. I think we're at that inflection point now. We're hearing about virtual cells and other stuff, but you need the actual cell data. And it's like, over the last 10 years, we've had billions go into AI drug design companies. They've had 3 molecules and they've all failed clinical trials, and that's just tech bros. They have hype. So I think AI will help us get maybe some of the routine tasks done, so we can actually focus on solving the problem, the creative side, and then, that may be where it'll all come in. But I don't know. Definitely.
AI, it's a tool, right? So it's great to have this tool, but it's not, it's not solving the problem, or it's not going to solve the problem. So it's a tool that we can use.
Thinking to the therapies, AI could be used as a tool, as clearly a tool, but with the incredible power. For example, there is a fantastic tool they are start using, in which they have on one side the whole amount of drug compounds available right now, even from the FDA approved, either on the market or not in the market. And on the other side, they are combining with the pathogenetic mechanisms. So they are pulling all together, and with the highly sophisticated algorithms and the AI, they are trying to see if and which drugs could be the right target for disease. And this is something which could be, it's a tool, but it could be extremely useful. And the second part is what I've already mentioned, the possibility to use real world data, as all of you, to generate synthetic jet data, to increase the amount of information to run a clinical trial, because otherwise the number in many cases would be too low. And it works. Again, it's a tool. And it's a tool on one side, and the other side you need anyway real data, because you can generate something from.
And when for the families, when Paolo referred to real world data, it means, and correct me if I'm mistaken, it means to get data for sure from the various interviews that we did, not only genotype, phenotype, not only the neuropsychologist interview, not only the occupational therapy, not only the speech therapy, but also to participate to RayRex, that maybe we have the platform where we can put in data, and ultimately from there we are able to provide data upon needed. And I would add a bit on top of that. I think it's very useful. Marco Tartaglia is not here. He is unfortunately sick at home, but he was able to do something. He will recover the session remotely. Of course, all of us will be invited, and he will speak about the methylation profile of CHAMP1. That being said, he was able to move in a rather fast way, meaning in some months he was able to do everything, because thanks to the families that donated blood sample to, in this case, Children's Hospital of Philadelphia, and then the collaboration, we come back to collaboration, of Children's Hospital of Philadelphia, we were able to ship the blood to Marcella, who did then the first step. I don't know, I cannot say technically what was it, but the first step, and then collaborate with Marco. You see, it was a circle. Families, CHOP, Marcella, Marco. This is the circle that we need to always try to improve and continue to push on? What was it the?
Yeah, that's true. It's not only families, but it's data also.
Sure.
Because I know for sure that Marco found extremely interesting data results. We are on the phone, also for white Saturn patients. But some are not, they don't fit with the others. And in this case, it's mandatory to have any kind of clinical and genetic information.
No. At this moment, we must be grateful to Daniele, Enrico, and Jeff, because. Sorry. No. Merit, everyone in the CHAMP1 Foundation, because we collected DNA samples from a lot of families, and for each patient we have detailed clinical information thanks to
The organization.
And of course, this is important also for testing the effect of any therapy in individual patients. Of course, we must start from baseline and then to verify which areas can improve. So I think that people working in the CHAMP1 Foundation do work very well, and thanks to you.
So, from now on, we kindly ask you to refer to CHAMP1 Research Alliance with the logo of Research Alliance. Thank you very much. That's very appreciated.
And thanks to Daniele for this excellent organization. Thank you.
Not only me, again, not only me. No worries. So, do we still have time? No. No? Okay. One last question?
It's time for a last question. Come on. Last question. Okay. So, Marcella, you mentioned neuroplasticity before, and I wanted to ask to which extension this neuroplasticity might improve the condition, and at which point of the therapy working on neuroplasticity could be done.
I have not the answer, of course, because I think nobody in this room can answer your question, but we know that neurons can undergo plasticity also in adult life, no? So, but I have, of course, I have not the right answer at this moment.
Thank you very much to everyone. I would say a final, let's say, thank you. We need to say that. Maybe you know, maybe not, we have, let's say, a scientific advisory board. And there is one guy who is, of course, must be the chair of the advisory board, and he's the one sitting with the blue t-shirt, the only one sitting with the blue t-shirt. So we will just to say to Stefano, and of course to extend it to the full advisory board, thank you very much for the additional job on top that you're doing for us. Thank you, Stefano. So,
it's time to go. The appointment is upstairs.
Like that. Ciao. Ciao. See you.
I'm not the one for, for the speeches. I'm just one who push people. It's you as the president.
So, as a president of the European Foundation, but as also member of, especially as a member. And come here, take a mic, please. As a member of the Research Alliance, we are very grateful to every one of you who participated so far. Look forward to do something tomorrow morning. I still don't know what. We will see. And with that, we would really like to say a very big thank you to you all, because otherwise we will be like just here sitting and probably crying one on the shoulder of each other. So thank you very much.
And I think the same can go for the families. A lot of you have travelled a long way. You've taken your holiday to come here rather than going to a beach somewhere in the middle of summer. It's really appreciated, you being here, what you're doing, and I really hope it's been beneficial. You've made friends, you've made connections. It's all just been worth it. It definitely has been for us. So just thank you to all of you.
Thank you.
Those who brought us all here, our champs. Thanks to them, we are